Okay, we're going to get started. So this week I've gotten all of your choices for the TRACO cores, one of which is going to be at Anton's facility where they have the robotics. So we're pleased today to have Anton Simeonov talking. He's the acting scientific director of NCATS. The title, The Use of Advanced Technologies and Small Molecule Translational Research. Anton.
All right, thanks, Terry.
Thanks everyone for coming. Your video conference system is muted right now.
All right. So now that we have silence, we can continue. So a couple of introductory remarks. You will hear from me various versions of this example. The translational research is a team sport. Really takes multiple disciplines to advance basic research discoveries to really reach tangible improvements in human health. And in the talk today, you'll see examples of biologists working with chemists, informaticians, and so on. So keep this in the back of your mind. It's a very different type of game where you really need multiple areas of expertise coming together to advance these basic discoveries. So I'm going to be talking about high-throughput screening, and it helps to define some of these terms, or at least provide the operational definition. So I lost my screen here, so I'm going to turn this way. So small molecules, these are generally synthetic or naturally occurring substances that are usually below 1,000 of molecular weight. And of course, a lot of the traditional drugs that you see in use are small molecules. Of course, now we have more and more of the other flavors, proteins, gene therapies, cell therapies, and so on. So what is an assay then? Not every test that one runs in the lab can be called an assay. Assay usually can be repeated multiple times with very low variability, possesses adequate signal to background, and can be scaled down or miniaturized for repeat testing, which brings me to the next term, high-throughput screening, which is the repeat rapid execution of assay many times over to test compound library for activity against particular target being isolated protein, pathway, phenotypic readout, you name it. And a library is a collection of small molecules, typically over 100 small molecules, arrayed in different formats, usually microtiter plates, frequently dissolved in DMSO as a standard solvent, and this allows you to run these screens in parallel fashion, generating many data points, either simultaneously or over the course of a couple of hours or days. I'm going to also talk about new developments in the field, telling you about how we developed a high-throughput platform to test drug candidates for repurposing applications, for new indications, and also testing for advantageous drug-drug combinations, because, as you know, drug cocktails are now frequently used in managing, say, HIV, but how do you go about discovering novel drug-drug combinations rapidly? Last but not least, there's the whole aspect of toxicology. A lot of drugs fail because of toxicity. We're working in this area to develop better predictive methods to prioritize compounds, and by compounds in this case I mean both drugs, drug candidates, and also typical chemicals that exist in commerce, because for very few molecules out there, there is animal data on how toxic or safe they are. So the way we operate these projects is they're all collaborations. Again, translation is a team sport. We provide the technical expertise, knowledge about the process, knowledge about assays, high-throughput screening, but we do not pretend to know the biology or the particular disease that we're studying. That expertise comes from the outside, from the collaborators who bring to us their knowledge, their test system, and their downstream expertise as far as say, xenograft models or anything that's beyond the realm of initial high-throughput screening. So we're able to test things in massively parallel fashion. They span the entire range of reductionism, if you will, starting from the simplest system, isolated proteins, which can be enzymatic assays, protein-protein interactions, protein-DNA interactions, and so on, going towards pathways, looking at particular pathway through embedding a reporter downstream or by various means of staining and so on. And then last but not least, the most sort of complex view on the high-throughput level, which is the phenotypic assays or high-content screens, where you don't necessarily look at particular pathway. You many times don't know the target, but you're looking for a particular change in the cell, whether it's, say, translocation or other features. So screening can be done in various plate formats. These are really representative pictures of the three most common plate types. What we have set up in our place is really the most advanced technological version of high-throughput screening, which is the use of 1536-well plates. I'm not going to go through the math here, but you can easily see why it is important to adopt this technology for a lot of types of questions that we want to ask. We can only answer them or approach them by using the 1536-well plate technology. Using these other plate densities is going to lead to either very long screens lasting many months or consumption of precious reagents that will make the screen literally impossible, either too expensive or impossible to set up because of lack of, say, enough primary cells and so on. You're really shrinking these assays down to the volume of, say, 2 to 5 microliters per test, as opposed to a lot larger volumes here. And of course, you're handling a lot less plates per experiment. So all of this is integrated into this robotic system that is shown here. The orange arms move the plates around. They remove the lids from the plates and transport them from workstation to workstation in completely automated manner. There are carousels for the compound libraries available basically as random access type resources. We can incubate, say, for mammalian cell-based assays for a couple of days in controlled environment. We have various liquid dispensers and different types of plate readers. So once you hit go, so to speak, you're only limited by what reagents you have and how complex the protocol is. The software knows how to queue the plates and make sure that everything receives the same type of treatment. Every plate is treated exact same way without waiting or skipping steps and so on. So this is all nice, and other facilities have this technology. So when we were setting up the system back in 2005, we wanted to really advance the field regardless of disease area or assay methodology. So we acquired the system, but we were still not completely satisfied with the way the process worked. So really tried to improve the process by introducing something that is really intuitively very obvious, which is why not test every molecule in a dose-response fashion? Traditionally, high-throughput screening is performed at a single concentration of your library, whether it's 10 micromolar or 20 micromolar. Statistical cutoffs are applied to the data that is obtained from the screen, and one picks these hits, so-called hits, based on, say, either greater than 50% activity in whatever assay you have or other criteria. Spurious noise from a single experimental result is still clocked as a hit, and only later, when you reobtain that sample and retest it carefully, you realize that it was single-point noise, maybe piece of dust hit the plate, or liquid dispenser added different amount of reagent. So a lot of resources are wasted at that point. You also miss shallow curves and other peculiar responses. So we actually invested a lot of effort into developing this dose response-based screening, which we called quantitative high-throughput screening, because you really are adding the quantitation dimension to it, diluting the compound library across about 4 orders of magnitude of molar concentrations. This way, right out of the primary screen for the active compounds, you get actual dose-response curves, which you can fit through the Hill equation and arrive at Hill coefficients, EC50s, IC50s, and so on. You can delineate between nice, perfect curves with 2 asymptotes against curves that are partial or shallow, or have completely negative or null response with 1 outlier. You can pretty quickly, almost automatically, figure out whether something is spurious noise or something worth pursuing. You can also rank the compounds based on potency and begin to derive structure-activity relationships based on the whole relationship between activity derived from these curves and the chemical structures of those hits. So we also have developed a system to run screens in biosafety level 3 environment. So the robotic system I showed in the previous slide is only qualified to run assays up to BSL-2 level. In Cliff Barry's lab in Building 33, we set up a high-throughput screening system that allows us to run BSL-3 type infectious organisms. These protocols are more complicated. A lot of times they require longer incubation times on the order of several days. So the plate type in this system is only 384 well plate. We don't screen in 1536 well plates in this facility because, again, for example, for slow-growing parasites, you literally need protocols that are 5 to 7 to 20 days. Still, the system is capable of performing all kinds of complex steps like sealing plates, peeling plates, incubating, reading multiple times, and so on. We've used it to run pilot screens on mycobacterium, and are gearing up to run a MERS type screen on that facility. So even though it's not part of the ANCAT's infrastructure, we did help develop it, and we're using it. Another aspect of this whole thing is teaching these collaborators, people like you, what an assay is and how to think about configuring a good high-throughput screening assay. A band on a Western blot is not a screening assay, for example. Things that require cell sorting, they're not good screening assays, and so on. So a lot of this has sort of teaching aspect to it, and this all started about 10 years ago with us actually convincing the Eli Lilly lawyers to release their internal assay development manual. Initially, this was just a website with a couple of volunteers adding chapters to that manual and basically a web link pointing to the whole manual without having the ability to convert it to PDF or print it for copyright reasons. Now it's grown into an actual eBook with pretty large body of editors, and we're hoping to add new chapters to it that pertain to preclinical development and also in the future stem cell-based assays. So in addition to screening, this whole process of early discovery, coming up with a tool molecule, or in some cases you can call it a lead or drug candidate, requires that one perform medicinal chemistry optimization on one or more hits. So we have an integrated pipeline at NCATS that does that. We have medicinal chemistry in the building as part of the teams that we have. Again, going back to the notion that translation is a team sport. The purpose of medicinal chemistry is manifold, of course. One wants to improve the potency of the molecule against the initial target. This is an example of hemotype developed against ubiquitin-specific protease 1, USP1, dealing with the emerging field of protein ubiquitination control and as relates to DNA damage response, the Fanconi anemia pathway in this case. Medicinal chemistry is also required to improve in vitro or in vivo ADME properties of the compound, things like solubility, microsomal stability, cell permeability, properties that don't really relate to the affinity of that molecule towards the target, but obviously are very much needed in order for one to transition from a test tube experiment to, say, animal experiment or testing in humans. The molecule cannot be very unstable, such that you would have very short half-life. So modifications can be made to make it more stable, remove some metabolic liabilities, and so on. So I want to give you one quick example of area of biology we worked on for a while, to kind of take you through this journey of starting from understudied target category, finding a small molecule modulator to basically de-risk the category, to allow for certain experiments to be run, for hypotheses to be tested, and really where this journey took us in the end. So back around 4 or 5 years ago, there was very little known about the histone modifying or interacting proteins, and these are several categories loosely defined as writers, readers, and erasers of histone code. Writers being enzymes installing epigenetic marks like histone methyltransferases. Readers being domains that interact with certain methylation states on histone proteins. For example, recognizing H3K9 dimethyl versus trimethyl, and then recruiting other proteins to control transcription and other processes. So there are many examples here of subfamilies, bromodomains being probably the most prominent. And then the erasers are the enzymes that remove marks, such as histone deacetylases, very well studied enzymatic class, and histone demethylases that remove methyls from usually histones, histone tails on arginines and lysines. So back when we started working in this area, there were practically no screening assays for any of these. There was no Constellation Pharmaceuticals, there was no Epizyme, there was no Agios, any of these companies that work in this field or near this field. So we actually set up some of the first screening assays for all these categories and ran the first 1536 well-based screens. And I'm going to give you an example of the last category, the erasers. So specifically, we worked on Jumonji histone demethylases. So it's an enzyme class that catalyzes this pretty complex reaction that involves oxygen and other co-substrates. It's part of this large family of oxoglutarate-dependent oxygenases. By the way, the HIF-prolyl hydroxylases are also part of that large family. The important thing about this enzyme class is they take trimethylated lysines as substrates. No other histone demethylase class does that. There is another class, demethylases, the lysine-specific demethylases. LSD1 is a prominent member. It does not use trimethylated histone as substrate, starts from the lower degree of methylation. So again, pretty complex reaction. How do you set up a screen to find inhibitors of these enzymes? So we used a coupling system utilizing the fact that one of the products from this reaction, the methyl group from the substrate that is being removed, is converted to formaldehyde. So we coupled the reaction with formaldehyde dehydrogenase reaction that utilizes NAD as a cofactor or co-substrate, forming as one of the products NADH, which is fluorescent in the UV range. So we would use excess formaldehyde dehydrogenase, excess NAD, and capture any and all molecules of formaldehyde formed on the fly, in line, in real time, and follow the fluorescence as a time course type measurement. So here's what the protocol looks like. We were able to miniaturize this reaction to 4 microliters total. And really this very simple sort of diagram actually represents about 2 months worth of testing of what can be combined together and what needs to be kept separate, such that you can run this 24/7 without things precipitating, without enzyme degrading, and so on. We had to run pairwise combination compatibility tests, literally going over the course of several weeks to understand that no, you cannot mix, say, the first 2 components with the enzyme, that you have to keep certain things separate, and so on. Finally, the protocol is absolutely simple. We have enzyme, we add compounds, candidate inhibitors, and then we start the reaction by adding everything else, which we could combine. Really nice, streamlined protocol, which allows you to run things in a parallel fashion, uninterrupted for a long time. And then, again, we collect fluorescent data on the fly. So back in the day when we ran the screen, we had about 250,000 compounds, again, all tested in dose response. We followed the screen, got a lot of hits actually, followed the screen with a lot of informatics analysis looking at autofluorescence, promiscuous type structures among the hits, and also had several experimental hit validation steps, including looking at the inhibitory properties of these hits using mass spec assay, which doesn't utilize any enzymatic coupling or any other tagging or labeling of the system. So among the validated inhibitors was this 5-carboxyate hydroxyquinoline structure. It was submicromolar inhibitor against the various isoforms within the Jumonji-demethylase family, but it was only weakly active against the other oxyglutarate-dependent oxygenases. So it was pan-active within the family, but selective for demethylases. We collaborated with the Structural Genomics Consortium back then to derive crystal structure to really enable us to understand the mode of binding for this molecule and therefore make better analogs. And we also had initial data through transient transfection type immunofluorescence experiment that showed that the molecule was active in the cell, inhibiting the demethylase catalytic activity of the target and not, say, transcription or anything else. So it was acting on target and not through some spurious side effect. So further metchem effort, making well over 100 analogs of this initial molecule resulted in this improved analog with better in vitro ADME properties. And of course, we published most of this early work And again, the early stories surrounding epigenetics were mostly related to cancer. This molecule has been sent to about a dozen labs worldwide to test in various cancer models. But what is interesting is the unintended use of this molecule. So it's a great tool molecule to study the effects of histone demethylation inhibition in cells. So it was purely by chance that we collaborated with an intramural investigator here on campus, Thomas Christie from the Infectious Disease Institute, to test a new biological hypothesis, which is that maybe through affecting the host histone demethylation machinery, one can affect viral infection. So Thomas Christie's lab has studied herpes virus and related viruses, and they've determined that this is a DNA virus which, upon infection, forms chromatin-like structures. Its DNA is compacted and, of course, carries the repressive histone marks like H3K9 trimethyl. And then, in turn, the virus uses the host demethylation machinery to remove those repressive marks, to affect demethylation, and therefore to reactivate itself from latency or to proceed through with productive infection and transcription of its immediate early genes and so on. So we wanted to test whether the histone demethylase inhibitors we had would be effective in herpes virus infection. Because again, unlike replication inhibitors, these would be acting upstream at an earlier stage of infection and hopefully would be more potent or more effective in stopping the infection or slowing it down. And this is indeed what happened. Adding the Jumonji-demethylase inhibitor to cells infected with herpes simplex virus 1 leads to a reduction in transcription of exactly the immediate early genes as expected from this hypothesis of utilization of the host machinery. Treatment with the inhibitor leads to dramatic reduction in viral yield based on looking at viral counts, also looking at plaque assay, leading to almost complete eradication of the viral plaques. By comparison, we have acyclovir, which is the standard of treatment, and it's a replication inhibitor. So you can see the Jumonji inhibitor performs just as well, if not a little better. These are images that are kind of hard to follow, but on the left side we have study that shows the inhibitory effect of the compound on infection that is progressing in suppressing the continuation of an infection. So you want to follow green here, where treatment with the compound leads to practically no green, and acyclovir still leaves some green spots, which are staining for the replication protein from the virus, and vehicle, of course, leaves it with a lot of green. Basically, the infection is progressing pretty rapidly. On the right side, we have an ex vivo experiment where mice infected with herpes virus are allowed to finish with the initial stage of their infection. The infection enters latency. When mouse ganglia are taken out of these animals and transplanted into culture medium, the latency is released. There is basically a reactivation process starting, and one, in this case, can stain in red for the viral proteins again. So in this case, we're looking at the effect of either acyclovir or the Jumonji inhibitor on preventing or suppressing the reactivation from latency. And It's hard to follow these. These are stainings for neurons, individual ones or clusters, but the count is shown here. And you can see that treatment with the demethylase inhibitor, both in single or cluster neurons, lead to almost complete reduction or suppression of reactivation from latency, whereas treatment with acyclovir is not as effective, even though acyclovir is used at twice the concentration of the Jumonji inhibitor. So again, this is in concert with the whole notion that we're acting upstream, preventing the reactivation, whereas acyclovir basically acts on the replication portion of the viral life cycle. So we don't have the illusion that this molecule will become a drug. It has certain chemical liabilities and other features that may not make it very desirable for further clinical development, but it was really a nice tool to show that this is a viable hypothesis, which is that affecting the host cellular machinery can be used to attenuate viral infection, something that people couldn't really do beyond the genetic means or siRNA means until such small molecule tool was available. So you never know where the journey may take you. We started with the notion of just designing the first inhibitors for Jumonji-Demethylases, looking at mostly cancer applications, and we have a very interesting story with potential antiviral application. I'm going to switch gears and talk a little bit about another route, right? One can take large libraries. At NCATS, we have about 500,000 such compounds in the library that are diverse and all that, and embark on medicinal chemistry after screening and hope one day for an FDA approval of the drug candidate. But one can also start with approved drugs and look for whether they can be used to treat a different indication, the so-called notion of drug repurposing. There are many such success stories of existing drugs that are finding new uses, but how do you really go about systematically discovering new uses for existing drugs? Well, there are two components to it. One is knowing what the drugs are, how many are out there, and then assembling a physical collection of those and testing it. I'm not going to dwell on this, but it took about 5 years for our informatics team to search databases worldwide, look at different languages, translations, deal with ambiguities in spelling, deal with ambiguities of chemical structures, redundant naming and all that, to find out how many small molecules have been approved for use in man. These include currently marketed drugs, but also investigational agents whose development has been stopped for, say, business reasons. This work was published some time ago, and you can get details from the paper, but there is also a browser associated with the paper where one can actually look up the drugs. And again, we're not talking just FDA drugs or European Union approved drugs. We also cover other major countries. Now, what is the number? There are almost 7,000 such agents. Again, not all of them are currently marketed drugs. And at NCATS, we have obtained about close to 3,600 of these. The first 1,000 or so are very easy to obtain. You can buy them as a collection from Sigma Aldrich. There's the NIH clinical collection. There's the Hopkins drug collection. These are all no more than 1,100 to 1,200 molecules. The remaining 2,000 or so, some of them had to be synthesized in-house or through CROs, literally being obtained on one-by-one sort of basis. So it's really a nice, precious collection, and we've screened it against more than 50 models of various diseases, including rare diseases and neglected tropical infections. Now, this is one part of repurposing, building the collection, building a unique collection, comprehensive collection of drugs. The other aspect is using combinations of drugs. Again, finding advantageous combinations, there is nothing new in this, but it's mostly being done through rational experimentation, starting from knowledge of a pathway and looking at molecules that may affect it and trying to combine them with, say, favorite oncology drug and so on. How do you go about doing it systematically on a large scale? We needed a platform for this, and again, there is dual use in this. One is to find better combinations that have higher efficacy, carry less toxicity due to lowering of individual drugs doses, but then results from combination studies can lead to new hypotheses as to why certain pathways conspire to give you synergism. You may not arrive at a particular novel drug-drug combinations, but you may arrive at a novel biology-biology combination or pathway combination. So running these screens on a massive scale can be doubly beneficial, if you will. So there is, again, several components to this. We need high-value library of small molecules, either drugs or something we call MIPE, mechanistic investigation plates. So these are libraries of small molecules that have not been in man, they haven't been through preclinical studies, but are very selective based on extensive testing. So we find those molecules literally through looking at papers almost manually and examining the quality of the data and trying to obtain these molecules either by in-house synthesis or again, targeted procurement. The other aspect to it is to find a way to plate these drug-drug combinations, to deliver these combinations in a high-throughput manner. And of course, last but not least, how do you analyze the data? You need a platform to make sense of all this data. So we start these by screening combinations of two agents, both dosed in certain range, applying acoustic dispense technology, which I will show you in a moment. And we follow these screens by running the hits that are most synergistic as 11 by 11 or 12 by 12 concentration matrices, really obtaining high resolution information on potential synergism. So this is the technology we're using. We're using acoustic dispense technology where the acoustic energy is applied to a well that contains solution of the small molecule, fires up a droplet that flies upwards and lands into a recipient plate. The droplets have literally picoliter sizes, and you can literally print on the bottom of the plate different doses of agent A and B. This lower right point showing the 0 point, so there is no drug there, there is only DMSO vehicle. And then along these axes, on the ends, the drugs are dosed just by themselves, and then in the middle you have the combinations of both agents. And really this allows us to print these really in high throughput. See if I can mute this. The system is completely contactless, so the recipient is on top here, and this is not a plate, it's just a plate lid for demonstration purposes. The source is the black plate on the bottom, and then of course the recipient plate is turned right side up and can be processed further by adding cells to it. And I hope you can see the spotting here, which in the real case of matrix or combination screening would represent the two agents at particular molarities. And we can customize this range versus that range to be different depending on the single agent potency. And we can do this on the time span of, say, 2-3 days. We can test one oncology drug against all approved drugs, several thousand combinations in rapid succession. The Informatics platform allows us to look at various indices of synergism. Some of you may know there is no perfect way to calculate synergy. There are many models out there. We can rank hits based on various indices, and we can also look at the actual heat map data on the fly. So this was all developed in-house recently. And if you want to know more about the platform, this is a paper we published back in the winter with Lou Stout and colleagues on some of the first screens run using this platform. We're expanding the platform now to go beyond oncology type applications, building specific libraries for infectious diseases and later on immunology and other areas. Because again, depending on the test system, you may not be able to screen everything against everything. So you kind of have to keep it confined, and by necessity, you have to build custom libraries or subsets to run those experiments. Last topic I want to visit is something a little bit more orthogonal to the drug discovery process or the process of running a screen, performing medicinal chemistry, optimizing a molecule, and then proceeding to, say, animal and clinical testing. And it is actually a pretty big problem, which is toxicity of molecules. While you're preparing a molecule for ultimate clinical application, you're mostly optimizing basic properties of the molecule against the target of interest or route of delivery and so on. How do you optimize for lower toxicity? It's something that is really hard to predict and test for, especially in high throughput manner, and clearly remains a problem. A related problem as pertains to toxicity is the fact that from environmental standpoint, from exposure standpoint, there are tens of thousands of chemicals generated by chemical industry that exist in commerce that are part of everyday materials and or everyday lives that don't have any toxicology data associated with them because nobody required the manufacturer to generate such data. Running all these tens of thousands of chemicals against traditional methods for toxicity assessment like long-term rat tox or other studies would be prohibitively expensive. So one way to look at this problem is to really think how to break down the problem of toxicity into component questions or pathways and apply high-throughput methods to be able to assess the thousands or tens of thousands of chemicals. Or another way to look at it is the organism, human or animal, is exposed to this chemical. Chemical goes into various tissues, organs, acts on various molecular pathways, targets within the cell, and beyond homeostasis, eventually something breaks and you end up with a toxicity outcome, also referred to as adverse outcome. So wouldn't it be nice to deconstruct the rat into various cell-based assays, biochemical assays, high-content assays, generate all that data using preferably cheaper assay methods, high-throughput methods, and then computationally build the rat back together or human. So this is the goal of a program that we started a couple of years ago called the TOX21. it's actually a consortium of NCATs in collaboration with the Environmental Protection Agency, another NIH institute, NIHS down in North Carolina, their National Toxicology Program, and FDA, where we started a couple of years ago and are literally generating data on various adverse outcome pathways, combining the expertise of the different agencies, where, of course, NCATS brings to the table the expertise in screening, assay miniaturization, and the other partners obviously contribute other types of expertise. On the EPA, especially there are large computational toxicology teams that look at the results generated by our robotic screening to begin to build these predictive models. This was done in a couple of phases. Initially, we just wanted to get the processes worked out, so small library of about 1,400 chemicals was tested sort of semi-manually against various assays. We've now graduated to a library of about 10,000 chemicals. These are all tested as 15-point dose responses in triplicate, generating really high-density data, very accurate data, redundant data deliberately, because unlike regular high-throughput screening where you just want a couple of hits and then you hope to progress them for drug development, here negative data are just as important. Most of the early assays run against this collection are sort of the low-hanging fruit, the so-called most important assays for toxicology, the assays for endocrine disruption, estrogen receptor, and related. And we're always on the lookout for new assays that relate to toxicology. So if you have ideas of assays that can be run on this system, by all means, let us know. We accept new assay proposals all the time. And again, we have various stress response related assays, and we'll be moving towards high content and other systems. Needless to say, this is again done robotically using different dedicated robotic system and incorporating various plate readers, including the acoustic dispense technology and other innovations. So to date, the program has been very successful. We've recently finished depositing the really largest data set of well over 50 million data points. Again, all dose responses on the 10,000 member library against the first series of assays. And again, we don't pretend to know everything. We're actually using the entire community worldwide to crowdsource the data analysis and model building. What do we mean by that? We provide data on the 10,000 compounds, including their structures, activity against the assays we've released in PubChem, and we're asking the community to build predictive models, and we test those models on a couple hundred compounds which we haven't released the data for. So that's what we're using to score these models, and the challenge actually is scheduled to end in a couple of weeks, and we have well over 2,000 models submitted for the various data sets and activity models. And we're also working closely with other non-US government agencies and nonprofits to expand those efforts. And this is just an example of one of the first papers dealing with these large screens on the estrogen receptor, which is very important system in toxicology. So we screened it 4 different ways using beta-lactamase reporter, luciferase reporter, full-length receptor, ligand-binding domain. So really 4 different versions. All of these initial data are compared in this paper. With this, I will end and be happy to answer any questions. So, so NCATS has, like most institutes, two faces, two sides to it. I'm on the intramural side. We're the people who run experiments, run screens, and work with outside collaborators to take their models, bring them in-house, and essentially set them up for screening. NCATS also has the extramural component where there are grants going out to various applicants to do work in the rare disease area. So on the intramural side, we have activities that span the entire spectrum. We work with disease foundations to set up new assays, to design new assays for rare genetic diseases, for example, or tropical infections. Sometimes this takes 2-3 years to generate new cell line, to embed new reporters, to really carefully look at the biology of the rare disease, whether it's silenced gene or lysosomal storage disorder that is caused by a point mutant. There are really very different flavors of rare diseases, right? And they're rare cancers, of course. We run screens on those assays. We also have a lot of drug repurposing efforts, and some of these are also taking place on the extramural side, where NCATS is funding clinical trials using molecules from pharmaceutical companies that have been basically paused. The company has started developing the molecule and then at one point has decided to stop the development, say for business reasons, they ran out of money or whatever, but otherwise there is nothing wrong with the molecule. Several companies have agreed to provide these molecules for repurposing trials. So essentially the pharma company provides the active ingredient, including placebo if needed. The outside applicant provides the model system and the rationale for why this, say, diabetes drug candidate should be repurposed for something else. NCATS performs the review and funds the clinical trial. So it's really a three-party sort of collaboration. And it took several years to design the master agreement because there are a lot of such relationships out there where academician is testing pharma company's molecule for something, but it usually takes 4 or 5 years to reach an agreement between University X and Pharma Y. So in this case, really the big achievement in our view has been to harmonize the agreement and really bring the parties together and literally tell them you have only a couple of weeks to finalize and sign, or else you're not getting this grant. So that's really the carrot stick there. So we have efforts on both sides of the table, if you will. We have our own internal screening efforts, and we work with a lot of disease foundations and individual academic investigators, but also on the extramural side, there is this sort of targeted repurposing effort through partnership with pharmaceutical companies. So, you know, when you have herpes virus, it stays silent and latent, and every once in a while you get the cold sore, right? And you put something on, and that ointment is usually acyclovir containing. So you're trying to basically reduce that outburst of infection reactivation through application of replication inhibitor. We think that by applying a Jumonji inhibitor instead, we would be more effective in reducing that reactivation, stopping it earlier. So the hope is to utilize this mechanism as a superior to replication inhibition in this case. But of course, demethylation of histones is really wide-ranging process, you're not going to be able to apply Jumonji inhibitor on a chronic basis. So it would have to be either for a cancer application where you know that particular cancer is dependent on high activity of Jumonji demethylase, or in the case of this herpes virus reactivation, which literally would require you to use this drug probably twice within 1 day, and you're done, ideally, because things happen so rapidly, and then the infection is over because it enters latency again. So you're not going to eradicate herpes virus from the human host. That's not going to happen because it's already there, but the goal would be to control the reactivation a lot more efficiently. Thank you. You guys taking a break? Go straight. Thank you. That's fine. Yeah.
Have a good trip. Okay, our next speaker is Sonia Jacquelou, and for background purposes, she got her PhD at the Rutgers University. Then she conducted a postdoctoral fellowship in France. And she came back to NIH, and she started working with Michael Sporn, who is famous for his investigations on transforming growth factor beta. So Sonia, her title is Transforming Growth Factor Beta in Lung Tumorigenesis. She's currently in the Center for Cancer Training. And she has an office just across the hall from me.
Pocket. Okay. Okay, great. Okay, thank you. Well, thank you, Terry. It's happy to be here to speak to you. And today I'll be talking to you about transforming growth factor beta in lung tumorigenesis. And lung cancer has become a very important disease in the United States. This year, lung cancer has become the most common cause of cancer deaths in both men and women in this country. There have been over 220,000 new cases that have been diagnosed this year among both men and women, and over 159,000 new deaths due to this disease. Most cases now occur in ex-smokers. However, there's a climbing rise in the number of cases that are among people who've never smoked before. And the 5-year survival rate is still a dismal less than 15%. Now, my laboratory became interested in transforming growth factor beta, or TGF-beta for short, very early on. TGF-beta is a multifunctional regulator of cell growth. It's a potent inhibitor of the proliferation of most normal epithelial cells, particularly in culture. It shows widespread tissue expression and has been shown to play a pivotal role in maintaining epithelial homeostasis. TGF-beta has also been shown to be associated with various types of cancers, including human lung cancer. And it shows context-dependent inhibition or stimulation of cell proliferation and neoplastic transformation among numerous cell types. So TGF-beta is a very attractive candidate for new therapeutic intervention approaches. if we can find them. So TGF-beta actually has its roots in another protein growth factor called sarcoma growth factor. Sarcoma growth factor is a polypeptide that was found to be secreted by murine sarcoma virus transformed mouse fibroblasts that could stimulate normal rat fibroblasts to form colonies in soft agar. And this was called a transformation assay that DeLarco and Todaro devised in the early 70s and used to discover this growth factor. Two classes of TGF have been isolated from Maloney sarcoma virus transformed cells. One class was shown to be able to compete with epidermal growth factor, EGF, for receptor binding, and this class was called TGF-alpha. Another class was found to not compete for EGF binding, but to be able to form colonies that could be enhanced by the application of EGF. And this class was called TGF-beta. So actually, sarcoma growth factor is actually composed of two separate growth factors that are genetically distinct, TGF-alpha and TGF-beta. And this was published by Robertson Sporn in the early 80s. Following the purification of TGF-beta became published from human platelets and human placenta, as well as bovine kidney. Again, all this was done at the NCI. To give you some idea of the purification, this is from bovine kidney. Bovine kidneys were typically obtained from the slaughterhouse and extracted with 8 liters of, typical prep was extracted with 8 liters of acid ethanol. Then after extraction, the debris was centrifuged out, and the extract was then left to precipitate with ether ethanol overnight in the cold room. The next day, the precipitate was redissolved in acetic acid and then applied to a 80-liter biogel P60 column for purification. 1 liter fractions were collected, and these were then lyophilized and redissolved for further chromatography purification. So final yield was typically 6 micrograms of TGF-beta1 protein. And this resulted in a 230,000-fold purification. So to give you some idea of the enormity of the columns that were used at that point, this slide shows the P60 columns that were used, and there's a 55-gallon drum in the right side of the slide that was used for collection purposes. So the assay for TGF-beta involved the growth of normal rat kidney cells in soft agar. Typically, a mixture of media, serum, NRK cells, EGF, and the sample was applied to an agar base and allowed to incubate for 1 week at 37 degrees with CO2. After 1 week, the plates were stained, and the colonies were counted in an Omnicon image analysis system, and I have a shot of one of the image analyzers that was used. So if no TGF-beta is present on the left side of the slide, you wouldn't see any colonies formed and be counted. If TGF-beta is present in the sample, then colonies would form and the bigger ones would be counted. So this slide shows a typical final HPLC purification of TGF-beta protein. And it shows that there is one band migrating at about 25,000 molecular weight, single band on a polyacrylamide gel. And Drs. Michael Sporn and Anita Roberts are credited with giving birth, so to speak, to the TGF-beta molecule. protein. And following the purification of TGF-beta protein, the amino acid sequence of the protein was determined, and it was determined that TGF-beta existed initially as a pre-pro TGF-beta form of 391 amino acids containing a signal peptide shown in red at the left-hand side of the slide at the carboxy terminus, with a latency-associated peptide or LAP shown in blue in the middle portion of the molecule, and the mature TGF-beta polypeptide consisting of 112 amino acids at the carboxy terminus. The crystal structure of TGF-beta was established in the early 90s, and it was shown to consist of two identical monomeric chains that were held together by an interchange disulfide bond with a hydrophobic pocket in the center portion. Now, TGF-beta is actually a member of what we call the TGF-beta superfamily. This superfamily consists of the 5 isoforms of TGF-beta, TGF-beta 1, 2, 3, 4, and 5, as well as the bone morphogenetic proteins and growth and differentiation factors, or BMP GDFs, and as well as the activin and inhibins. So it's a very large family of cousin proteins. Now, transforming growth factor beta per se is a 25,000 molecular weight disulfide bonded homodimer. 3 highly homologous isoforms have been shown to exist in mammals. These are TGF-beta 1, 2, and 3. In addition, TGF-beta 4 has been identified in birds, and it has highest homology to mammalian TGF-beta 1. And TGF-beta 5 have been identified in amphibian, and that has highest homology to TGF-beta 1 as well. Most recently, a 6th TGF-beta isoform has been identified in fish, and again, that shows most homology to TGF-beta 1 in mammals. The principal sources of TGF-beta are platelets, bone, and spleen, and most cells are known to express TGF-beta and its receptors. TGF-beta is usually secreted in what is called a latent inactive form that must be activated in order for TGF-beta to perform its various functions. And the superfamily of TGF-beta contains not only TGFs, but a number of other cousin proteins as well. The members of the TGF-beta family act as central control modules for many important biological processes. These include development, immune system function, reproduction, angiogenesis, aging, tissue repair in response to injury, metabolic regulation, and proliferative homeostasis. So whatever process you can imagine, TGF-beta has some role to play in that process. Major biological responses that are regulated by TGF-beta include the inhibition of cell proliferation, regulation of apoptosis, differentiation, and immune cell function. TGF-beta also stimulates the accumulation of extracellular matrix and can promote chemotaxis. The current model for TGF-beta signaling pathway involves the receptor proteins and the SMAD proteins, and this is referred to as the TGF-beta SMAD-dependent pathway because it depends on the existence of the SMAD proteins. So typically what happens is, I'll have to move around here, the type 2 TGF-beta receptor is constitutively phosphorylated. It binds the TGF-beta ligand, and the ligand can be either the TGF-beta isoform or BMP or GDF, whatever. This complex then recruits the type 1 TGF-beta receptor to make another more enhanced complex, and the type 2 receptor transphosphorylates the type 1 receptor. Once this happens, then, this complex is able to then interact with the SMAD proteins. It depends on whether you're talking about TGF-beta activin or VMPs, whether this will bind to the SMAD proteins, SMAD2 and 3, or SMAD1, 5, and 8. These are the SMAD protein, the SMAD receptor proteins. And once the signal is transduced here, the receptor SMADs are able to then interact with the common SMAD4, and common SMAD4 has the ability to translocate the signal to the nucleus, where various transcriptional events that TGF-beta plays a role in can take place. Now, the whole process can be circumvented by the inhibitory SMADs, SMAD7 or SMAD6. Now, clinically, TGF-beta has shown to have tumor suppressor activity. For example, germline mutations in the TGF-beta pathway components have been shown to cause familial predisposition to cancer. And this has been shown to occur with SMAD4 in juvenile polyposis syndrome. The TGF-beta pathway components have been shown to be somatically mutated or deleted in some human cancers. And these include the TGF-beta type 2 receptor in human non-polyposis colorectal cancer, as well as SMAD4 being predominantly mutated, deleted in pancreatic cancer. And thirdly, the reduced expression of TGF-beta signaling pathway components or overexpression of endogenous pathway inhibitors have been shown to be associated with disease progression. And this involves the type 1 and type 2 TGF-beta receptors, as well as SMAD7 and a TGF-beta associated proteins called SCI, which I won't have time to talk about this afternoon. Clinically, TGF-beta has also been shown to have tumor promoter activities. For example, TGF-beta1 has been shown to be elevated in many advanced human tumors and has been correlated with metastasis or poor prognosis of cancer progression. And these include such tumors as breast, colon, stomach, liver, etc., as well as the lung. And shown in the lower right side of the slide is a prostatic adenocarcinoma, which has been immunostained for TGF-beta1. And here we see the brown staining for TGF-beta, indicating that it is at the interface between the tumor parenchyma and the microenvironment. So, what is the role of TGF-beta in carcinogenesis? Is it a hero or is it a villain? Well, it turns out it's both. TGF-beta is a proximal effector of the malignant phenotype. It's also a potent growth inhibitor and tumor suppressor under some cases. And it's also a pro-metastatic factor in other cases. So how do we unify all this? Well, there's a current unifying hypothesis which uses the ability of TGF-beta to switch from being a tumor suppressor to a tumor promoter pro-oncogenic factor during cancer progression. And how does it do this? Well, in normal epithelium, say in the lung, TGF-beta, the tumor suppressor activities of TGF-beta predominate. And TGF-beta acts as a tumor suppressor. But as changes in the genetic and epigenetic context take place, TGF-beta loses the responsiveness to TGF-beta is lost, and the expression and activation of TGF-beta goes up as metastasis increases. So in metastatic cancer, the pro-oncogenic activities of TGF-beta begin to predominate over the tumor suppressor activities, and this increases as tumor genesis increases. Now, I told you about the SMAD-dependent pathways for TGF-beta. There are also SMAD-independent pathways that exist for TGF-beta, and these include such pathways as the TAC-MEK1 junk pathway, the TAC-MEK1 P38 MAP kinase pathway, the RAS-ERK MAP kinase pathway, the RO-ROCK pathway, and the PP2A S6K pathway. So these are independent of the function of the SMAD proteins. Okay? So why is this important? Well, we became particularly important in the RAS MAP kinase pathway because in lung cancer, the KRAS gene shows an activational mutation in as many as 50% of human lung adenocarcinomas. Mutation of even one allele of KRAS has been shown to increase the appearance of lung lesions in human cancer. There is valid crosstalk between SMAD-dependent pathway and the RAS-MEK signaling pathway as well. Activation of the RAS pathway can modulate TGF-beta1 signaling through the SMADs or without the SMADs. And in vitro studies have shown that TGF-beta1 can dominate over the mitogenic effects of RAS, but that activated RAS can override the interproliferative effect of TGF-beta1. So this is shown here in this slide, the various pathways that can go wrong with TGF-beta being a tumor promoter. So if the RAS MAP kinase pathway is activated, then the tumor suppressor activities of TGF-beta can theoretically be circumvented to allow tumor promotion to predominate. So the broad goal of my laboratory, one of the broad goals of my laboratory was to determine the role of TGF-beta in the development and malignant transformation of lung epithelial cells. And this work was performed in the epithelial carcinogenesis section of the Cell and Cancer Biology branch of the CCR of NCI. Our objectives were threefold. First, to examine the effect of TGF-beta1 deletion and KRAS mutation alone and in combination on lung tumor incidence and pathology. Second, to determine early events in the development of lung lesion and their progression. And third, to identify potential signaling transduction pathway changes that increased with increasing tumor genesis. For our work, we employed 4 mouse models: the AJ mouse, the C57 Black 6 TGF-beta1 heterozygous mouse, and I'll refer to this as HET for simplicity, the AJ Black 6 TGF-beta1 HET mouse, and the TGF-beta1 HET KRAS latent activatable mouse. We asked 2 questions. Does lung tumor genesis affect the TGF-beta signaling pathway? And the reciprocal question, does the TGF-beta signaling pathway affect lung tumor genesis? We employed the AJ mouse model first because this mouse model is known to be very susceptible to chemically induced lung tumors using such carcinogens as ethyl carbamate or benzpyrene. The tumors develop in a time-dependent fashion, and they go through stages of hyperplasia, adenoma, and carcinomas, similar to the human lung cancer progression. The carcinomas that appear are histologically similar to human lung adenocarcinomas, and very importantly, the same molecular mutations occur in both human and mouse lung tumors. such as overexpression of KRAS and loss of p53 expression. Now, the carcinogen ethyl carbamate goes to at least 2 different types of metabolic events. The first is a detoxification pathway, which employs simple hydrolysis using esterase and water to produce ethanol, CO2, and ammonia. There's also a bioactivation pathway that utilizes CYP2E1 to form vinyl carbamate intermediate, which is then metabolized to vinyl carbamate epoxide, and it is this epoxide molecule that is able to bind to various macromolecules and proceed with carcinogenesis. So our standard protocol was to inject 2-month-old aging mice with ethyl carbamate and then sacrifice groups of mice at various monthly intervals up to 12 months. Now, shown in this slide is an immunohistochemical staining for TGF-beta 1 and the type 1 and type 2 receptor protein in lung tumors that formed after 2 months, 4 months, and 8 months of ethyl carbamate treatment. One can see a brown staining for the TGF-beta 1 protein as well as the type 1 receptor protein at all stages of tumorigenesis. In contrast, expression of the type 2 receptor for TGF-beta is decreased, reduced in comparison to the other 2 proteins. So this slide shows this a little better in comparing staining, immunostaining for the type 1 receptor with the type 2 receptor. And one sees very reduced expression of the type 2 receptor in the lung tumor compared to the type 1 receptor. Okay? In comparison, staining for the normal bronchiole in both proteins is almost identical. We also looked at the expression of the mRNAs for the type 1 and type 2 receptor. Interestingly, we show loss of the type 2 receptor mRNA in the PCC4 cell line, which is a mouse cell line that was derived from after treatment with ethyl carbamate. So we show decreased production of the protein and mRNA for the type 2 TGF-beta receptor in the aging mouse model system we were using. We also treated these mice with benzpyrene and also another lung carcinogen, and showed that essentially we see both immunohistochemical staining for the type 1 receptor and TGF-beta ligand and hybridization in situ hybridization to the mRNAs for these proteins in the tumors. In comparison, the expression of the protein and message for the type 2 receptor is reduced in the tumors, while there's normal expression, equal expression of these proteins in the surrounding normal bronchioles. So our model shows that TGF-beta can be compromised by the reduction of the type 2 receptor protein, which again leads to decreased ability of the TGF-beta molecule to act as a tumor suppressor and more to act as a tumor promoter in this lung system. Our next question was, does deletion of TGF-beta 1 affect lung tumorigenesis? For this question, we use the C57 Black 6 TGF-beta 1 mouse. Now, the TGF-beta 1 knockout mouse was generated in the C57 Black 6 strain in the 90s. And the mouse is born and seems to thrive like its litter mate. It's a bit smaller than the normal litter mate, but at about 21 days, something begins to happen. It starts to succumb to what we call a general wasting phenomena, and shortly thereafter dies. So the knockout mouse is not a good mouse to employ to use to study lung tumorigenesis. But the TGF-beta1 heterozygous mouse, it seems to thrive and reproduce and has no problems. So we treated the TGF-beta1 heterozygous mouse with the carcinogen diethylnitrosamine to look at liver tumorigenesis initially. And we found that there's increased incidence of liver tumorigenesis in the heterozygous mice compared to the wild type. Interestingly, we also observed that there was increased lung tumors in the het mice compared to the wild type mice, and that was very unexpected. So in order to better look at this in lung tumors for lung tumorigenesis, we then crossed the AJ mouse, which was wild type for TGF-beta, with the TGF-beta1 het mouse in the C57 Black 6 strain to generate the AJ Black 6 F1 generation, which included genotypes that were heterozygous and wild type for TGF-beta1. And our plan was to treat these mice with carcinogen and look at the lung tumors that were generated. And shown here is a immunohistochemical and in situ hybridization for the TGF-beta1 protein. And here we show evidence that our system, we wanted to be sure that our system was working, so the heterozygous shows decreased staining for the TGF-beta1 protein in the het compared to the wild type, and decreased expression of the TGF-beta1 message in the heterozygous mouse compared to the wild type mouse. Okay? We also showed that with northern blotting, there's decreased expression of the TGF-beta1 protein in the heterozygous mouse lung compared to the wild type mouse. And we also showed the same thing with using competitive RT-PCR in the heterozygous mouse compared to the wild type. So we proceeded as before. We injected the 2-month-old mice with ethyl carbamate and sacrificed mice of the 2 genotypes at monthly intervals for up to a year. And essentially, we looked at hyperplasia, adenoma, and carcinomas that form. And we showed that in all 3 types of tumor stages, we showed increased tumor incidence and multiplicity in the TGF-beta1 heterozygous mouse, shown in red, compared to the wild-type litter mates, shown in green. And there was also decreased tumor latency, especially in the carcinomas that form. While carcinomas started forming at 4 months in the heterozygous mouse, it took almost 12 months before we see carcinomas forming in the wild-type mouse litter mate. So we looked at the expression of the type 2 receptor protein in these lung lesions, and shown at the bottom right with the pink arrows, you see decreased expression of the type 2 TGF-beta receptor in tumors of the het mice compared to the wild-type carcinoma shown on the left. We also looked at the relative levels of type 2 receptor mRNA in the lesions from the TGF-beta1 heterozygous mouse lung that have been treated with the carcinogen ethyl carbamate. with increasing tumorigenesis. And we were able to see decreasing amounts and levels of the type 2 receptor mRNA with increasing lung tumorigenesis as tumorigenesis proceeded from hyperplasia to adenoma and then to carcinoma. Our next question was, does deletion of TGF-beta1 and mutation of KRAS affect lung tumorigenesis in combination? For this, we use the TGF-beta1 heterozygous KRAS latent activatable mouse. The KRAS latent activatable mouse we obtained from Tyler Jackson from MIT. To study the interplay of these two genes, we generated the TGF-beta1 KRAS latent activatable mouse by crossing the TGF-beta1 heterozygous mouse in C57 black 6 background with the KRAS latent activatable mouse in the SV129 background. We then generated 4 types of mice. The TGF-beta1 heterozygous KRAS latent activatable double mutant, the TGF-beta1 wild type KRAS latent activatable single mutant, the TGF-beta1 heterozygous single mutant, and the wild type mouse littermate as well. So to show you what we saw at the levels of histological pathology, these are lungs that were taken from 6-month-old mice after treatment. And we see pearly white nodules that are the lung tumors in the mice with the latent activatable KRAS genotype. We saw only very few spontaneous tumors appearing in the non-latent activatable genotypes. We looked at the effect of TGF-beta1 gene deletion and KRAS mutation on mouse survival with these 4 mice we generated. And we were able to show that there was definite significant decrease in the lifespans in the heterozygous latent activatable mouse and the wild-type latent activatable mice compared to those mice that didn't contain the KRAS mutation. We looked at the pathology of the lung lesions that formed in these mice, and we saw increased hyperplasia and adenoma in the wild-type latent activatable mouse, shown in green, compared to the latent activatable het mouse, as well as increased carcinomas in the heterozygous mouse, shown in red, compared to the wild-type latent activatable mouse. We stained for the TGF-beta1 and ligand in the type 2 receptor proteins, and shown with the pink arrows, we see reduced expression of the TGF-beta1 protein as well as the type 2 receptor protein in the heterozygous latent activatable adenocarcinomas. Now, we also looked at the expression of the various SMAD proteins and using Western blot analysis. And here shown typically is comparison of the wild-type latent activatable with heterozygous latent activatable. We see expedited reduction of the type 2 receptor and increased production of the SMAD3 protein in the het latent activatable mice by about 1 month compared to the wild-type latent activatable mouse littermate, which the other genotypes showed virtually no change in these proteins. Now, rather than show you several Western blots, I put this all together for you. So essentially, using Western blot analysis, we see in the TGF-beta1 heterozygous KRAS latent activatable mouse expedited reduction of the type 2 receptor protein and expedited production of the SMAD3 protein. But we see reduced production of both the SMAD4 and SMAD7 proteins. We also looked, because we were interested in the RAS pathway, we also looked at RAS and RAF1 expression, and both these proteins showed expedited increase in their production by about a month each time. We also conducted real-time RT-PCR and showed that there were reduced expression of SMADs 2, 3, 4, and 7 in the adenomas, but reduced expression of the type 2 receptor in SMADs in carcinomas. So not the same thing is going on at all stages of carcinogenesis. It seems to be a very complex system that we're trying to sort out. Now, we also looked at the apoptotic index in these mice, and we showed that there's reduced apoptosis, reduced level of apoptosis in the latent activatable adenomas compared to the wild-type latent activatable adenomas. So in our system, using these various mouse models, we're able to show that there are several ways in which the TGF-beta signaling pathway can be impacted in order to turn a known tumor suppressor more into a lung tumor promoter. We showed that the type 2 receptor can be decreased, leading to lung tumor promotion. Activated RAS-MAP kinase can lead to promotion of lung tumors as well. Decreased expression of SMAD4 also leads to lung tumor promotion, as well as compromised apoptosis leading to lung tumor promotion. So it's possible for carcinogens to counteract the activity of what's known to be a good tumor suppressor to make it really a tumor promoter, a powerful tumor promoter. So I'd like to acknowledge the people that were involved in this study. In my lab, Jerry Anderson, he was a very good postback, and postdocs Yang Kang, Alina Nomova, Jyotsna Pandey, and Sarah First, and they've gone on to better and higher positions as well. And I'd like to again acknowledge Tyler Jacks for the KRAS latent activatable mouse. And I'd be happy to answer any questions if you have any. You can't all be overwhelmed. Okay, one question that I get from some people is, where's this work going now? Well, unfortunately, I've now moved into the extramural field, and I monitor and manage a number of different grants for NCI. And there are various people, investigators in the extramural scientific community who've taken up this work and are pursuing it. So it's a project that I think has a lot of legs and can be morphed into various projects on its own. So, any questions? Yes. I'm sorry, can't hear you. Yes, there are some therapeutics that are being worked on. Unfortunately, they're very toxic. So that's the problem here, okay? There are a number of small molecules that are being tested, but so far they've not been of much use due to toxicity and due to they're not very easy to manufacture as well, okay, unfortunately, okay. Okay, thank you. You get out early. Enjoy the weather.